Barbara here at Maverix Biomics, and I appreciate the opportunity to give you an introduction to the Maverix Analytic platform as well as our integration with IPA. So first I'd like to go over what the data set is that we're using for this case study for our demo today. This is a publication by Wamstead et al. It's a Cell paper, and in this study they were characterizing cardiomyocyte differentiation in vitro. So what they did is they took mouse embryonic stem cells and they followed those through differentiation to beating cardiomyocytes. And then what they did is interrogate the transcriptome so that they could look at gene expression as it was measured both at the start, looking at the embryonic stem cells. Also, samples were taken at 2 intermediate stages. And so those different samples were enriched for mesodermal cells as well as cardiac precursor cells. And then at the very end, they collected a final sample, which was the cardiomyocytes at the end point of that cardiac lineage. So some of the key observations they noticed was a decrease over time in the embryonic character of the cells, increasing to the cardiac nature of the final cardiomyocytes. And they also found some key upstream regulators that were activated during the differentiation process, which drive downstream events. And so as you can see in this slide, we're starting with the embryonic stem cells. That's one of the samples that we have in our data set. The mesodermal cells, cardiac precursor cells, and cardiomyocytes. And for each of those, they noted some marker genes. And for each of these different cell types, these marker genes will be important because we're going to actually take a look at those in our final data set. So I'd like to take you now to the Maverix Analytic platform. What you should be seeing here is the dashboard. So anybody that has an account on the Maverix Analytic platform, this is the sort of main landing page when you log into your account. And what I'd like to do is give you an orientation to the platform, how to navigate around, and then I will go through an example of how to upload data and configure and launch an analysis on the platform. And then we'll go through some of the results and outputs from that analysis, as well as the data visualizations that you can use to explore your data on the platform. And then the final part will be to show the ease of our one-click solution to upload and transfer that data for analysis in IPA. So to begin with, I'll start here on the dashboard. As you can see here on the left-hand side, we have different projects. These are different projects that are associated with this account. And all of the data on the platform is actually organized by project. So as you see me clicking through these different projects on the left-hand side, you'll notice that the content on the right-hand side will change with that. And as well as being on the dashboard organized by project, the same is true for the main sections within the platform, which you can see up at the top. This is our main navigation bar, and you can transfer between the dashboard, the data section, the Explore tab, and the Results section. And all of those, the data that's included on there, as well as the processing, is organized by project. So right now I've selected this main project here, which is where our case study exists. And you can see that these shortcuts here at the top, these are sort of bookmarked sections within the platform where you can jump immediately to some of your higher level outputs and results from the analysis. The activities pane down here gives you some information on notifications from the platform, for instance, when you've started an analysis within that project, as well as when an analysis has completed. Up here at the top, you can see that there's also a section where you can easily jump between projects. And this is really nice when you're not on the dashboard, because once you're off of the dashboard, you won't have this ease of navigating between the projects here on the left-hand side, but you can always do that from this project menu here. And then one final thing, just in terms of main navigation, if you go to the account settings, this is where you can see the information about the account that you have. So for this account that I'm in, I have 2 panes here on the left-hand side, and this is where I could go to change my password, to log out of my account. And if you're a user that has access to more than one account, you'll be able to see those different accounts here, and you can switch between them. If I select this platform demo account here, this is an account where I'm actually an admin within the account. And now you'll see 2 additional panes on the right-hand side, and this is where I can manage user roles. So I have the ability from this section to revoke and grant admin permissions to other users within the account, as well as remove users and add them. So if I was to click in this text box here next to invite a user, input a colleague or collaborator's email address and click the invite button, this would invite them to my account, and they would get a separate email where they could register to the platform and select their own secure password. And now they would have access to this account, and you could collaborate together within this account and look at the different analyses and results. So going back to my dashboard, I'm going to demonstrate what these different sections within the platform look like. If I select the data tab, you'll see that for this initial project, there's not a lot of data in there. And this is where I was saying if you switch between the projects using this menu here, now you can see the file library and all the files that are associated with that particular project. You can sort this table by create date as well as the data type and any of these other columns. And this is a nice way to be able to quickly get to some of the data that you might have that's derived from your analysis. So this project file library contains both files that you would have uploaded for analysis as well as the outputs from that analysis. And selecting any row within that table will allow you to click and download that file directly to your computer. So if you wanted to look at these BAM files, these are actually deployed, and I'm going to show you this in a moment, as genome browser tracks within the integrated genome browser that's available within your account. But if you did want to download these and look at them off the platform, it's very easy for you to download those files from here. The next main section within the platform is the Explore tab. And this is where you would see any of the analyses that you had run within this particular project. You can see here a list of the different analyses that were run as well as their status. So you can see these all have a green check mark and they're complete. And pretty soon, once we launch an analysis, you'll see an example of what one looks like when it's running. This is also where you would start a new analysis. You would go up to this right-hand corner and click Start New Analysis, and we're going to go over that in just a moment. The final section is the Results tab. And for each different analysis, whatever the final results and outputs are, this is where you would find some of those higher level outputs. So for example, a differential expression analysis has cummerbund as the outputs from the cuff-diff part of that analysis. And this is where you would find those cummerbund outputs. And this is a really nice place when you want to collaborate with somebody else who's using your account, because for each individual plot that's available from the results, you can leave notes on that that are specific to just that one piece of data. So you can see here that someone has left a note on this, and somebody else can leave an additional note for that, or another member within that account who has access can also just leave a comment. And that's an easy way to be able to collaborate and make notations about the different results from your output. So what I'd like to do now is show you how easy it is to upload data to the platform, configure, and launch an analysis. So we start by going back to the Data tab, and we click Upload Files and select Add Files. And then from here, you can just select those files off of your computer. Once those files are queued, if you notice that you've made any errors and you've got something queued up here that you don't want, you can click Cancel and remove that. Once those are all uploaded and ready to go here in the queue, you can just click Start Upload. There's one limitation, and it's actually a limitation of the browser interface. If you have any individual file that's larger than 4 gigabytes, then that's a file that you can't upload from within this interface here in the web browser. But in those instances, that's where we would actually set up a user account for you so that you had direct access to the S3 bucket in the Amazon cloud, and you could upload files directly in batch as well as download files from your S3 bucket once the analyses are complete. In this example, I'm going to return to the project file library here, and these are the files that I've uploaded for this example of how to configure and launch an analysis. So the next step would be to go to the Explore tab, and from here I would click Start New Analysis. From here, I would select the analysis that I want to run, and this analysis kit that I'm selecting here is the mRNA-seq for differential expression. This is an analysis overview that gives a description of the analysis, includes the configuration pane here, which we're going to come back to. Additionally, we provide some details of the analysis, which includes references, as well as a list of the bioinformatics tools that are used for this analysis. We include here the versions for each of those, And each of the analysis kits that we have available to run from the platform that's pre-configured has a version of its own. And so that in that way that we can keep track of all of the different tools that were used for that, the different versions of those tools, so that if you did come back to the platform and wanted to repeat an experiment or run a new set of data using the exact same analysis, we would be able to make sure that you had loaded and were configuring and running the exact same version of the analysis kit on the platform. Finally, we include here some benchmark data, as well as the analysis overview, which lists the main modules and steps within the analysis, as well as a description of them and some of the outputs that you would expect when you're running that analysis. So what I'd like to do now is show you an example of how to set up an analysis here. We're going to configure one, and this will give you an example of how simple it is. You would give your analysis a name, and this would be something that you would be able to view from the analysis execution history. So you can just give it a label that makes it easy for you to find it later. For the next step, you just click Add Samples, and I'm going to add my first sample. So this one's going to be my embryonic stem cell sample. And then I click Add FASTQ files. The files that I uploaded are paired end reads, so I'm going to select that here. And then I can begin adding my replicates. So for this first sample, since I have paired end reads, I have to add these as pairs. If you had single end reads, then you could come to this kind of a pane. You would select single end run from the previous screen, and you could add all of your replicates at the same time. For paired end reads, you do select them 2 at a time. So then I'll add my second replicate here. So now for our embryonic stem cell sample, we have 2 separate replicates that are loaded here. The only other thing that you need to select is clicking this checkbox on the right-hand side if your reads come from a stranded library. So if you do have a stranded library, you check here. If it's unstranded, that's all you need to do. And now I have my first sample. Then I'll just go and add my second sample. And then add the FASTQ files for this sample. There's my first replicate. And the second one. And now I have my 2 samples loaded. For the data set that I went over with you, you saw in the case study there's actually 4 different samples that we would have and replicates for each of those. For this example, I'm just going to load embryonic stem cells and the cardiomyocytes, which is the final cell type in the lineage. And now there's only a few other options that I need to select. So I would come here and I would select which sample I want to use as a control. For this pre-configured analysis on our platform, we actually do the differential expression using 2 separate tools. We use CUFDIF and we also use DE-seq. For the configuration here, CUFDIF does require that you select a control sample, and there could be instances where you really don't have a specific control, but you do need to select one of the samples to be the reference sample at this stage of configuration. For the mapper, I have an option of selecting between 2 different de novo splice aligners. I can select Top Hat or Star. Top Hat is a gold standard, very highly cited. Star is another tool that we offer as an option here, which can actually improve the overall runtime of your analysis. It can cut it down by half. And so if this was something that you wanted to select, you would have that option here. So I'm going to go ahead and select Top Hat. For the platform, you select the platform that your reads came from. If they're Illumina or Ion Torrent, you would select that here. And then you would select the organism that you wanted to use as the reference genome for the mapping step. So in this case, there are some instances here where we're selecting a reference genome from an organism where there's only 1 genome assembly that's being offered here. For mouse, we actually have the option of selecting between 2 different genome assemblies. And it's possible that I had previously run some of this data against a past genome assembly, and I might want to select MM9 here. So that's what I'm going to go ahead and do for this configuration. And then the final thing that you need to pick is whether there was a spiking control in your sequencing run. Once that's done, you just click Launch Sample. And now your analysis is launched. And you can see here now that the very top analysis in my analysis execution history has a status of running, and this is the one that we just launched. So I'm going to go ahead and click on that and show you what it looks like as it's first starting. So for each step here, you can see that if you click on them, you see the same thing that you saw in the analysis overview with a brief description of what that step is and the different types of things that are happening within that step, as well as the outputs that you would get. If you click on each step, you can also see the details. So for analysis configuration, what you're really looking at is all the inputs that you just selected for this analysis. And you can go back to this later on as a reference to make sure that you selected the correct genome, to make sure that you're using the mapping tool that you thought you were selecting, as well as looking to see which control sample you had selected. If you look over here on the left-hand side, you can see that our analysis configuration step is completed. It has a green check mark. And now we're already into the quality assessment step. Since this analysis just kicked off and is running, I'm going to actually take you to one that's completed. And this is using the full data set from our case study. So once again, as you look at each of these individual steps that has completed, you have the option of clicking to look at details or the outputs. If you click details, you can actually take a look at the command line and see the bioinformatics tools, the open source tools that were used, as well as the parameters that were set at the command line. And then if you click output, you can start taking a look at some of the outputs from that step. So for quality assessment, we use FASTQ. Clicking on any of these will show you the larger version of that plot. So for this one, we're actually looking at the before trimming and filtering for this particular replicate. And what we do is we do FASTQ with the raw reads, and then we do adapter trimming, and we do quality filtering. And then once that's complete, we redo the FASTQ so that you can actually get a look at the before and after for each sample. So you can see these have really nice high quality scores even before trimming. But if you did have a data set that maybe had some adapters in it or some low quality reads, this is a step where you can actually filter those things out. And after the trimming and filtering, that optimized set of reads is what will move through into the next step of the analysis, which is read alignment. Once again, if you click details, you can see down here, this is the command line, but you're additionally getting some notifications from the platform about what's being published to your account. In this case, the BAM files that are some of the outputs from the read alignment step are being deployed to the genome browser as genome browser tracks, and those will be available in the integrated genome browser when you're looking at the data visualizations. Finally, the differential expression step, we can take a look at some of those outputs. These are the outputs that I was showing you on the results tab. So these are some of the ones that actually get promoted to that final results tab because they're some of the final outputs from your analysis. And once again, if you click on any of these, you can take a look at them in more detail. You can download the entire CuffDiff output file. And then if you scroll all the way to the bottom, you can see that in this case, we also have the DE-seq output file down here at the bottom. And so the two different analyses are run. You have access to download all of the output files from both of those. In addition, you can take a look at the heat maps, and this is a really nice way to start looking at your data and doing some discovery and sort of narrowing it down to look at the data set that you're most interested in. So what you can see here is an example of one of our data visualizations on the platform. On the left-hand side is a heat map. And on the right-hand side is the integrated UCSC genome browser. As you mouse over the heat map and see any of the individual rows, clicking on one of those rows will load that gene and its locus into the integrated genome browser, and you can start exploring some of that data. You can see here that you're seeing the read coverage tracks from the analysis, and you can view that in the context of public data sets that are available in the UCSC genome browser. And you do have the ability to go in and configure this from here. You can go to this menu, you can configure both the density of the tracks that you're seeing. You could add additional public data sets that are available. And so you do have the ability to look at all of that right within the, your account so that you're actually looking at your private data in that context. And there's a lot of different ways that you can look at this data in terms of configuration. For the heat map itself, some people might like to look at a heat map, and other people might want to view that same data in a table view, and so we do offer that option as well. If you go up to the left-hand corner and click the down arrow, you can see that you can toggle between the heat map view and the table view. In this instance, this works a lot like the heat map in the sense that if you do click on any of these rows, that will load within the integrated genome browser below. And in the table view, you have the option of sorting on any of these different columns, whatever makes it easiest to look at your data. You can sort on abundance, you can sort on fold change, and as well as p-values. So that's a nice way to be able to explore the data in the platform. So I'm going to toggle back to the heat map view. Another thing that I'll say is that using the same menu here, you can actually download any of the data that's in the current data set that you're viewing in the heat map. So what I'd like to do is show you some of the ways you can change those settings so that you're zeroing in on the subset of data that you want to view from your analysis. So in the settings menu, what we have in the top is the sample pane, and from here you can select the control that you want to compare all other samples to in your heat map or your table view. So when I was discussing the differential expression tools that we're using for the analysis, I did mention that DE-seq doesn't require you to select a control, and this is an example of how you can view the data and customize it based on the DE-seq analysis. DE-seq does an all versus all pairwise comparison between all samples, and at this stage you have the choice of selecting any of the samples that can be used as the control, and then within your heat map you would be looking at all other samples compared to that one sample. So right now we have it selected for embryonic stem cells. You also have the ability to reorder the rows within that heat map. The most customization that you can get here is if you were to select custom from this menu, and now you have the ability to remove samples. You can add additional samples. For instance, if you wanted to look at cardiomyocytes versus embryonic stem cells, this pane here for customization lets you actually select the control sample for each sample being viewed in the heat map individually. So now I can look at one versus embryonic stem cells, a second row maybe versus the precursors, and in order to be able to distinguish those, this is something that you could do in terms of updating the column labels here. You have the ability to change any of these, and then once you click apply, it's going to apply all those changes and re-render the heat map with those changes intact. In addition, you have the ability to sort your heat map. So the sorting right now is hierarchical clustering, and you do have the ability to change that and sort the heat map using chromosome, p-value, abundance, or the fold change. Once you click apply, those would be applied and the heat map would be re-rendered. This is an example of some of the filters. The current heat map that we're looking at has two different filters set on it. So one is for p-value with the minimum value less than or equal to 0.001. I've also included a log2 fold change absolute value setting here to filter my heat map, and I've set that to greater than or equal to 2. If you change any of these values and click apply, that's going to re-render the map on this other side, and you can see that you now have a smaller number of genes within your heat map for you to view. I do want to show you a couple of more things. The gene list is a really nice way to be able to zero in on some data that you might be interested in. So I've already uploaded a gene list here. If I select that, I can add that to my heat map, and it supplies some markers next to each of those genes within the view that you can locate them. To upload a gene list, you just create a list of genes, and this is just a small text file that includes just the gene symbols. Once you open this, it'll load it here, and all of the genes are being recognized within this data set. Any of them that wouldn't have been would have shown up in red. But either way, those are things that you can load and then use later. So these are the list of genes that were in that initial slide that I showed you for the case study, where they were looking at the genes that were marker genes for the different cell types within the cardiac lineage. If I add this, click apply, and then go back and take a look at my heat map, I can actually see these genes that they've been marked in my heat map. So now I can, as I scroll through the heat map, I can actually take a look at any genes that are associated with those different lists, and that'll be noted in the tooltip here so that they're easy to identify. Another way that you can do that is once you've clicked on and added some of these gene lists here, you can sort by them. That's available here. That'll sort them to the top of the list. So now when you're looking at your heat map, you can see them listed here. And you also have the ability to filter based on those so that you could actually limit the genes that are being displayed within your heat map to just the genes that are on these different lists. So I just wanted to show you very briefly what these genes that were considered marker genes look like in this heat map here. So these are some of the ones that were noted as being available at the point for embryonic stem cells here. And then as we move down through this list, you can see some of the ones that were with the cardiomyocytes. And so being able to look at these here, you can see the differential expression across these just by viewing in the heat map. And if you were to download this data set, you would be able to take a look at some additional information with them with regard to the abundances as well as the fold change between those samples. And this is all visible here. You can see that this was one of the genes that was later, and you can see that the embryonic stem cells actually have fairly low abundance, and that's increasing in that instance there for the mesodermal cells. And so you can view that data, and that's a good way for you to be able to look at the data set and compare it to what you saw in a publication and then go from there. So what I'd like to show you now is, of course, one of the main things here was our integration with IPA. One of the simple things we have is a link right here in the tooltip. This will take you to the gene view at Ingenuity where you can get some more information on that gene. This opens in a separate window, and you can start to take a look at some of that data, find out more about that gene, some gene ontology terms, as well as functional information. But now that I have this data set filtered, what I'm going to do is go to my menu here, and all I have to do is one click, and what this is going to do is load this data set to IPA and then analyze that data, which can then be viewed there to do a lot of the downstream analysis that's going to be the most interesting. So this data set that I have right here, you can see on the tooltip, is about almost 1,700 genes. You can upload larger sets of data if you want to, and then reanalyze them and filter them once they're available in IPA. But I'm going to go ahead and click to load and analyze this data, and it's going to take these 1,700 genes approximately that I had in my data set and pass those through to IPA. And then this is something that Stuart is going to take over from here, where he's going to go through and actually show you how you would go about analyzing this data, refiltering it, and doing some of the downstream analysis that's going to provide you a lot of the information about this, this data set and this set of differentially expressed genes from this case study. So here's IPA opening up. It's got my data set. It's going to upload it really quickly, and then from there what it'll do is start analyzing that. And you can see right here that I have some data sets that I've uploaded already, and I can start taking a look at the analyses, and these ones are currently running, and this is where Stuart can take over and tell you all about IPA and how you can look at your data there.
Great, thanks Barbara. Can you guys hear me and see my screen?
Yes, we can, Stuart.
Oh, excellent. Okay, I'm going to show some slides first, and at the end hope to have time to go back into IPA and demo some of this live. But it's a really seamless integration between the tools. So, but before I do that, I really do want to give an overview of Ingenuity and IPA a little bit, just for those people who don't know it as well as me. So let's see. So Ingenuity, the way we have created our tools to analyze gene expression data and other things is to have curated findings from the literature and third-party databases. So we have curators, PhD curators, who read papers and capture findings in a very systematic way using an ontology, so that those are computationally available to tools like IPA. So when you have data coming from a nice tool like Maverix upstream, you then can put it into IPA, and in our tools do the interpretation of the biology behind these changes in gene expression or in proteomics or metabolomics. So I wanted to give you a little bit of an overview of some of the things that IPA can do, because I can't cover everything in this talk. So for those of you who aren't familiar, we have these gene views, and this is what Barbara was alluding to. When you click a gene now in Maverix or in IPA, you can get to a web page that describes that gene or in a lot of detail. And on that gene view, we have a human isoform view, which shows the different splice variants for human genes at this point, as well as links to findings about specific isoforms. there's an interactive tool that can show you what protein domains are encoded in each of the exons, and it's a really nice way of looking at gene structure. We have canonical pathways, several hundred of them that are both metabolic and signaling and they're fully interactive, and they capture some nice, well-known biology. Some of the newer tools in IPA are what we call upstream regulators and causal networks, and these are tools to infer what's happening upstream of your data. What molecules like transcription factors or cytokines or microRNAs, whatever, have been activated or inhibited to explain the gene expression you're seeing in your data set. And that's upstream regulators, and causal networks takes it multi-levels to up even higher to link regulators together. I'll talk about that at the end. We also look downstream. What's happening predicted-wise to diseases and biological functions? Do you have increasing cell movement, or do you have decreasing proliferation, et cetera? It's a prediction based on the gene expression data, the directional gene expression data. Just as this looks upstream, this tool looks downstream. And then I'll talk about a new tool that's come out just this past weekend that integrates both of those together, which we call regulator effects. And it's putting the upstream regulators that are predicted, connecting through your data set to the downstream effects that happen. And that's, I think, a really powerful tool, and you'll see that in this cardiomyocyte study. We have tools for microRNA analysis as well. I won't be able to cover those today. I'll mention Bioprofiler briefly at the end if there's time, which is a search tool for connecting diseases and functions to genes and compounds. One of the best tools in IPA is being able to really interact with networks and understand how genes connect to compounds and what are their relationships to diseases and biomarkers. And this is sort of an interactive tool which you can explore biology this way. Okay, so just to emphasize how well used IPA is by the scientific community, we've reached our 10,000 milestone of papers that have published using IPA. So as of February, we have 10,000 papers that people have published using IPA. So we're really proud of how people use it and find value and publish with it. Okay, so you already saw this slide. I won't go through it anymore. But just to make it extra clear again, we're ratioing everything back to the starting culture. So mesoderm against embryonic stem cells, cardiac precursors against embryonic stem cells, et cetera. So there's going to be three what we call observations, or three data sets in a way that we're going to be analyzing. You've seen a nice demo of how this works, and it's simply just clicking that button and moving it to IPA seamlessly. And just to make it clear, what the data was filtered at a p-value of less than 0.01 and a log 2 fold change greater than or equal, absolute log fold change greater than 2.3, which is about 5-fold. So those are the two cutoffs used in IPA. And so we can ask things about what are the consequences of the gene expression changes, what happens trending over time, what's happening upstream, what's going on downstream, how do these connect? And I'm going to walk through each of these types of questions that we can ask. And you can also go on further and customize IPA content for that research question and do a more advanced analytics as well. And I'm going to cover those at the end. So what are the functional consequences? So this is a tool we call downstream effects analysis, and I showed you that each of these big squares are like a category of functions or diseases or phenotypes. We organize them in our ontology to these larger categories like cardiovascular system development. The color is the predicted effect on that functional category. So in each of the little squares is a particular functional category, and orange means predicted increase. So it looks like many cardiovascular system development functions are increasing they're increased. So for example, you can look down here in this table, which breaks down each of these into a table. So the contractility of cardiac muscle is predicted to be increased, and we use a z-score to computation for that prediction. I won't go into details now how that works, but positive z-scores greater than 2 are predicted to be increased, and negative z-scores less than -2 below -2 are considered decreases, the blue color. So there's decreased cardiovascular disease, there's increased cardiovascular system, muscle development is mostly increasing, there's some bone functions in here that are decreasing, cytoskeleton's being reorganized as over time. This is the cardiomyocytes. This is the last point in the development. But this is one of the situations. We're going to look at a comparison in a second. But you can, sorry, going back here, you can click on any one of these or more, any ones you want, and then do a display of network. And what it does is it now shows you the details. Here are the genes arranged around these functions that are from your data set, that drive the prediction. So this gene being upregulated in the data set is driving a prediction of decreased cardiomyopathy. It's also that same gene, because of the way the findings from the literature are arranged, essentially, that the same gene is driving a prediction of increased contractility of cardiac muscle. So this visualization allows you to see which genes are common or shared between two different or many more functions, and how they interact. And always you can double click on an edge here, a relationship here, and find out the literature behind it. So again, decreased cardiomyopathy, increased contractility. What's happening over time? Is there a way we can look at the pattern over time as these cells differentiate? So yes, you can. There's a tool called comparison analysis that lets you do that, a heat map of those predictions across the different conditions. So this is you can sort by, you can do hierarchical clustering, you do various sort methods. This is using a trend sorting. I'm just going to, so you notice these little sliders, you can just manually set these however you like, and I'm looking for an increasing trend over time towards cardiomyocyte differentiation, and it sorts the table, sorts the heat map to get you those. And so this is a zoom in on that, and so you see things like contractility increasing, heart rate increasing. These cardiomyocytes are actually beating in culture at the end. So then you can, of course, click on one of these like I did here, and it shows you this network again, so you can evaluate which genes are involved. So that's nice. A lot of increasing functions, and you can also look at the same kind of tool. You can look at pathways. So looking at same trend for pathways, you can see how glycogen degradation, there's two different glycogen degradation metabolic pathways are increasing in significance. This is not a Z-score, this is a significance, Fisher's exact test. And you can see how these pink ones are the enzymes involved in glycogen breakdown, and they're all upregulated in the data set, especially in the last one, which I've selected here. So you can see how, you can look at how the glycogen's changing, the energy source is being changed as these cells differentiate. But what's happening upstream? So you can do the same kind of analysis, upstream regulators, and do the trending. And this is actually filtered for transcription factors. Like I said, you can look at lots of different kinds of upstream molecules like microRNAs and kinases and all kinds of molecules. But this is filtered for transcription factors and set to look for things that are increasing over the differentiation course or decreasing in predicted activity. And you see lots of upstream molecules that are transcription factors, I should say, that have effects on cardiogenesis, differentiation. But to even go deeper, what is the biology? What is going on here? This is where this new tool that came out this weekend is really powerful. So how might those upstream regulators drive the downstream effects? So the way this tool works, the reason for the tool is it explains how a phenotype or function or disease can be regulated in the data set by upstream molecules. So it can be a great tool to find drug targets, or explain mechanisms, or discover unknown new regulated disease connections that that might not be known yet. And what I mean is something from here, an upstream regulator, down to a downstream effect. There might not be a known relationship, but your data set might be telling you that that's what's happening. This is a chemical to disease function. Well, let me go to the next slide. So let me talk about how this works. So it's simply integrating the two results, the upstream regulator analysis with the downstream effects analysis. Essentially, as I was showing you those networks, they're essentially a network around something. So an upstream regulator with its targets from your data set, or a disease or function with its with the targets from your data set. You can statistically merge those. So if there's a significant overlap by Fisher's exact test, you can integrate those two networks, and then you can essentially find this situation where an upstream regulator is driving a disease or function. And if the directionality is correct, in other words, like this gene is activated, and when that gene, or sorry, I should say upregulated, and when that gene's upregulated, that's known to increase the disease or function. Well, then this edge is causally consistent, and so we score for the ones where the edges or the relationships are causally consistent. Those score higher. And it does it multiple times, so you don't just stop with one. You add multiple upstream regulators, multiple diseases downstream. And if there's an existing relationship, like I was saying, there may well be a known connection between an upstream regulator and a particular disease function or phenotype. The algorithm will draw that connection, but it doesn't depend on that. That's sort of an annotation. It doesn't depend on that being existing. What's kind of cool also is you can customize the type of networks you get. Excuse me. So the default is to just find all upstream regulators that are genes, RNAs, or proteins. All upstream regulators of that type connect to any type of disease or function, and there is some cutoffs to make sure that the stuff you put in there is of the highest significance. But that's the default. But you can customize it. So you can say, well, I really just want transcription factors married to functions, or I really want a compound that can regulate functions, or I really want the simplest type, where it's just a one-to-one connection between a regulator and a function, or you can completely customize it. So these are just some sort of presets for you to answer different questions. So this would be a transcription factor and function example, or a compound as a regulator of function, or the minimal type, like I was saying, a one-to-one. Here's a microRNA predicted to be affecting migration of endothelial cells based on this data set. Okay, so the way the results are presented initially is in the form of a table, because there's multiple hypotheses or multiple networks that get generated, and so they're ranked by score, and you have essentially three columns that say, here are the regulators in this row, here are the regulators that connect through those targets to these diseases and functions, and here's if they're known or not. And it'll annotate whether so if it's at 0%, that would mean there was, like this one, there's absolutely no connection known yet between the upstream regulator and the function. And then the higher percentage ones are ones where it's more well known. And again, the consistency scores, as we call it, are higher for causally consistent and densely connected regulator effects networks. It's essentially clusters of regulators that have a similar activity on downstream effects cluster together in a network. So I've broken this into some pieces here so we can go walk through it. So here's a typical network. Here's the cardiomyocytes, the last stage, versus the embryonic stem cells. And there's a number of upstream regulators, most of which are predicted to be activated, one of which is predicted to be inhibited. And they connect through to the data set targets, from this data, and then again on through to the downstream effects. So contractility of muscle is predicted to be increased, hypertrophy is predicted to be, or cardiac fibrosis is predicted to be decreased. And this is just automatically connected for you to make a hypothesis for what's going on. And what's really nice is when you look at these, I mean, these are many of the key players in heart development, and they're connected to the functions that you'd expect. And you can, what's another neat thing about this is this is one way of looking at the result, but you can also just one click and show this in the form of a subcellular layout in IPA, and you get sort of an instant pathway. Here are in the extracellular space, you see growth factors and other molecules such as those, and then through receptors down into the cytoplasm and activating kinases, et cetera, and down into causing transcription factors to be activated or inhibited. So that's what in the nucleus here, and those are all driving different outputs that are shown over here. So it's sort of a quick way of finding a hypothesis that might explain what's going on. And one of these, this function, is something that we put in ourselves using a new tool which we call My Findings in IPA. This is a custom. This didn't exist in IPA until we added it manually, and this is something you can do yourself to customize IPA for your research. And so I just want to show you quickly how that works. So this is customizing IPA for your own research. We have call it My Findings. You have a way of uploading a batch of findings in a spreadsheet format. And we also, just this past weekend, we have a new tool right inside IPA. You can just draw yourself a relationship, and it becomes part of IPA for you and your people in your license for IPA. So your people on your license have access to these findings. They're sort of shared. And so this is how you do it in IPA. So let's pretend we are interested in also adding the question, hey, what's happening in induced pluripotent stem cells as well? I mean, we know they're embryonic stem cells, but what if I wanted to track mouse iPS cells and I found this paper that talks about generating cardiomyocytes from iPS cells? You could go and get that paper, and then you could add My Findings. You basically put this known gene on a pathway, and you create this from scratch. This is a proprietary node, and draw a relationship between them. So you simply just draw a line, and then you take this pop-up dialog, and you just annotate your GATA4. GATA4 is increasing causation in mouse of cardiomyocytes. And it creates this sentence for you. You just hit the little button, and it creates a sentence. You put in a PubMed ID, or it doesn't have to be published. It could be something internal to your company or to you. You just cite it and put in a project. So everything's organized by project, so that someone who's not looking at cardiomyocytes in your institution may not might turn that project off when they do an analysis. But if you're interested in that, you can include it. You submit it, and you create a new relationship. And within 24 hours, it becomes available to everyone on your institutional license. And the last thing, let's see how much time we have. I'll try to go pretty quickly here. You can also do what we call causal network analysis. So I showed you upstream regulators, which is simply looking at a regulator and its literature connections to known targets and then those are pulled from your data set. Causal networks can use a little bit higher power by putting together other regulators in between an upstream regulator. So we call this the master regulator. It can potentially activate or inhibit other regulators, which in turn create expression changes in your data set. So this is a sort of multi-layer analysis, and there doesn't have to be a direct connection between this master regulator and the data set. So there's a potential to find completely novel regulatory mechanisms through these master regulators. And you can also automatically score that master regulator against something you're looking for, like the phenotype or even another gene. So I'm trying to go quickly here. So this is doing the causal network analysis and doing a heat map of that. And you can see this molecule JDP2, which is predicted to be increasing over time. And this is the causal network. So this is the master regulator, and it talks to 3 other molecules, or this is a family of MAP kinases. And then in turn, those talk to your data set. And I haven't added the data set molecules yet, but there's 124 data set molecules that would sit down below these. So here's a multi-level hypothesis about JDP2. And in fact, if you go look at it, it's known to have histone modification roles and differentiation, but a role in cardiomyocytes has not been published before. But like I said, you could annotate. So I put in differentiation of cardiomyocytes and to automatically ask the question, how does JDP2 interact with differentiation of cardiomyocytes in some way? And in fact, it can through June and cyclin D1, I think that is, and this other molecule, and that's how it can drive increase of differentiation of cardiomyocytes through these other mechanisms. So it's predicted to be activated, predicted to drive differentiation. So this is a really powerful tool, especially for discovery, and being really comprehensive about looking upstream. And again, it doesn't have to be a gene as an upstream regulator. Here's a chemical which looks like it could drive increase of cardiomyocyte differentiation and through these intermediates. And here's the heat map showing that. And if you look at this molecule, it's actually interesting. It's known to increase cardiomyogenesis of mouse embryonic stem cells. So it may work through this mechanism to do that, but that was a known thing. Okay, and lastly, really quick, so we have time for questions. There's a new tool also called Bioprofile in IPA, which allows you to really, in a detailed way, filter to genes like causally associated with a particular disease or phenotype in a particular way, like which genes, when decreased or increased, cause a certain endpoint, what type of genetic evidence supports it, is it a knockout, and you can do this kind of analysis to say which molecules, when increased or decreased, have a causal effect on a particular endpoint like contractility of myocardium, and you get this nice table where you really can filter in a detailed way what species, how it's activating, how it's inhibiting, and really explore a set of genes in a detailed way, or compounds. It works for compounds as well.